Good morning, everybody, and welcome to the second day of the Kasha Research Conference. We will start with session 3, Zoonoses and One Health. And before starting, I want to briefly introduce the first speaker, which is Rachel Lowe. Rachel Lowe is a Royal Society Dorothy Hodgkin Fellow based at the London School of Hygiene and Tropical Medicine. Her research is supported by the Global Challenge Research Fund, and she did her PhD in mathematics at the University of Exeter. And besides mathematics, her research interests are climate change, planetary health, and infectious disease. Dr. Lowe is the director of the Landsat Countdown in Europe and transdisciplinary collaboration tracking progress on health and climate change. And in January, she will join the Barcelona Supercomputing Center as an ICREA research professor and global health resilience team leader. She will talk about climate and emerging infections, the present and future. And just before you start, Rachel, remind that you can write your questions during the session in the chat. So after each speaker finish the talk, we can translate the questions to her or him. Thank you very much. Rachel, the floor is yours.
Thank you, but bear with me 1 second. Thank you very much. Shall I share my screen?
Yes, please, if you can share. Yeah, we can see it.
Okay, thank you very much for having me today. It's a pleasure to take part in this session. So I'm going to be speaking about climate and emerging infections, present and future. And I'll be focusing particularly on mosquito-borne diseases, which is the area that my research focuses on. Talking a little bit about how we can track and understand the emerging infectious diseases and also project them into the future and think about adaptation solutions to those. So you may well be familiar with this visualization. It's known as the warming stripes. It's a graphic produced by Professor Ed Hawkins from the University of Reading. And each stripe shows the global annual temperature from 1850 through to present day and the projections until the end of the century, depending on the actions that will be taken over the coming years and decades. So we can see over the last 30 years, we've had a really dramatic warming of the global climate. And from this point on, if dramatic action were to be taken, then we could potentially limit warming below 1.5 degrees, although the commitments made at Kock were not very promising in that direction. And if we're to continue as business as usual, then we would find ourselves in a very worrying and dark space with temperatures well above 3 degrees C. And along with this recent warming over the last few decades, we've also seen an expansion of diseases like dengue fever. And in the 1970s, there were 9 countries reporting epidemics of the disease, and that number has now increased to over 120 countries, not only in the tropics and the subtropics, but also it's spreading to other areas. And the WHO estimates that around half the world's population is now at risk. So we recently conducted a modeling study where we projected the change in the length of the transmission season and the population at risk of the 2 most important mosquito-borne diseases, malaria and dengue. So we used a multi-model approach, and we found that if using the scenario where we continue the emissions as they are, that we would see an additional 4.7 billion people at risk of these diseases, whereas if we were to take drastic action to reduce warming below 1 degree C, that could be reduced to 2.4 billion. We also saw a general expansion to higher altitudes and also a northward shift of the transition zone in the United States and into Europe for dengue. So of course, climate change is not the only factor that can impact the risk of the transmission and outbreaks of infectious diseases. It's also attributable to population growth, unplanned urbanization, and inadequate infrastructure. This image here is one of the largest favelas in Rio de Janeiro, and you can see all the little blue patches on the screen. They are temporary water storage containers. And the residents of this favela are forced to rely on these containers for water during water shortages. And these, of course, provide the ideal breeding sites for the domesticated Aedes aegypti mosquito. And these kind of conditions, combined with extreme climatic events, can provide the perfect storm for explosive epidemics of dengue fever. And we know that the global spread is attributable to the increased volumes of international travel, trade in used tires, as well as the climate crisis, which is leading to more frequent and extreme events. So in our team, we are working to try and understand how we can make the most of all the Earth observations available to us from satellites, from drones and other monitoring devices, and combine this with disease surveillance data to co-create decision support systems with local decision makers who would like to build climate resilience to outbreaks of these infectious diseases. So we've been working, thanks to a UK Space Agency funded project with the public health authorities in Vietnam to develop an operational dengue early warning system, which provides probabilistic forecasts of the risk of dengue from 1 month to 6 months in advance for the different Vietnamese provinces by using seasonal climate forecasts from the UK Met Office, which give the evolution of the likely climate conditions 6 months ahead of time. So this is the platform that is available to the decision makers, and this is currently being used to help target resources, mosquito control and educational campaigns to those areas where it's most needed. We're also working with partners in the Caribbean to try and understand the changing epidemiology of dengue and the recent introduction of chikungunya and Zika viruses. And it was noticed that the dengue epidemiology pattern was changing given the increased frequency of droughts in the island. And Barbados is one of the most water scarce countries in the world, and it suffers from these climatic extremes, which are often linked to the El Niño southern oscillation. So we combined information measuring hydrometeorological extremes with dengue cases and temperature to try and understand how these extreme events might be interacting. And we found that drought led to an increased risk of dengue 4 to 5 months later, whereas exceptionally wet conditions led to an increased risk of dengue in the same month or 1 month later. And of course, warmer conditions several months before was also increased the risk. So we're now working with the Ministry of Health and with the Barbados Meteorological Services to monitor these hydrometeorological extremes and combine them into an early warning system. So if we observe hot and dry conditions followed by wet conditions, then our model would predict a high risk of a dengue outbreak. Whereas if we were to experience cool and dry conditions and it continued to be dry in the next season, then the overall risk of dengue would be low. So we're working to incorporate these different lead times into an early warning system at the moment. And this finding of the impacts of drought on dengue have been incorporated into the Caribbean Health Climatic Bulletin. In 2020, there was a regional drought, which was which coincided with a forecast of particularly wet and warm conditions. So a warning went out to the countries in the Eastern Caribbean to be prepared to control mosquito breeding in temporary water storage containers, which are relied upon during drought conditions in the region. We've also been working in Brazil for several years to work towards a dengue early warning system there. Ahead of the 2014 World Cup, we produced a dengue outlook for the Ministry of Health by combining seasonal forecasts and the latest dengue surveillance data available at the time of forecast into a model to produce probabilities of exceeding outbreak thresholds across the country. And we found that our model did a much better job than relying on seasonal averages alone. So this was the sort of first bit of evidence of the feasibility of a dengue early warning system in areas where dengue is endemic. We've been incorporating information about ecological zones, looking at how the seasonality and intraannual variability changes across the country. We have applied the modelling technique that we developed in Barbados, and we have found that we see the similar patterns where drought conditions 4 months ahead of time increase the risk of dengue. And when we looked at this across the wide geographical domain of Brazil, we saw that this risk is exacerbated in urban areas, and particularly areas which suffer from water storages. So this is useful guidance for control activities of how to prioritize control in urban areas versus rural areas, and when and where to be monitoring these extreme events. So you may well be familiar with the Landsat Countdown project, which is a multi-institutional collaboration tracking progress on health and climate change. And for a few years now, I've been involved in the climate sensitive disease working group. This is an example of the malaria indicator that was included in the 2021 report that was published last month. This is showing how climate suitability for malaria has been increasing over the last 70 years, particularly in highland areas, and this is particularly exacerbated in areas with a low human development index. So that is many parts of Africa we can see there. So these kind of tracking exercises give an indication of where policy should be directed to really make sure adaptation mitigation strategies are in place to deal with these increasing threats from climate change. And as Julia mentioned, I am now the director of the Landsat Countdown in Europe. So this is the newest member of the Landsat Countdown family. We published our framework paper in September 2021 in the Lancet Public Health. And the goal of this project is really to leverage the wealth of data available across Europe from the European Environment Agency, Eurostat, from Copernicus, and bring all this data together to produce region-specific, fine-scale and evidence-based indicators to inform policy across Europe. And we welcome collaborators to this collaboration, so please do get in touch if you would like to contribute an indicator to this effort. And we're currently developing our work with Professor Jan Semenza and Professor Joachim Rocklov to integrate this framework that's used in the IPCC of monitoring vulnerability, exposure, and hazards to climate change, and to incorporate this within a One Health framework where we are incorporating animal, environment, and human health in an integrated framework to make sure that we are prepared for an increase in zoonotic infectious diseases in Europe in the coming years. So here you have my contact details if you'd like to get in touch, and from January I will be based at the Barcelona Supercomputing Centre, so I will look forward to interacting with you from there. Thank you.
Thank you very much, Rachel, for this nice presentation. And meanwhile we wait for people to asking questions. Just a reminder, you have there a site where you can write your questions. I wanted to ask you some, taking into account, as I told you before, that I'm not an expert in modeling and in bioinformatics. So, well, one of the questions is when you do such models, how many variables you take into account? I mean, I guess you can add as much as you want, more or less, no? I mean, taking temperature, humidity, no? Or which are the criteria to add or exclude different variables?
Thanks for the question. Yeah, it really depends where we're working on what are the interests of our stakeholders. So if we're working, for example, in Barbados, we've been working on island-specific problems, or we're working with time series. So in that case, we've been exploring using distributed lag nonlinear models. So then we're looking at sort of multiple lag times with a particular variable of interest. For example, we've been using the standardized precipitation index to understand drought combined with a temperature indicator. Usually minimum temperature we find is the best variable. So when we're working in that kind of situation, it's very difficult to have other temporal drivers that we can predict in advance. So we try and keep the models as parsimonious as possible. When we're working in a spatio-temporal framework, so for example in Brazil, we consider other factors in the model that vary across space, like population density, urbanization, sanitation indicators like access to piped water, refuge collection. But it really depends at the spatial scale you're working. So if you're working at quite a coarse spatial scale, usually a lot of those socioeconomic variables are confounded by urbanization. So we're really trying to produce the most simple models so that you can make predictions either at sub-seasonal to seasonal timescales, or indeed use your models for projections into the future.
Mm-hmm. Yeah, I think this is very important, and it's a question that always when we are talking about this One Health approach, no, we discuss it's not the same, no, to find solutions depending on which country, for example, we are talking about. So, well, I have more, but first I will move to the chat. There is a question from Carmen Aguilar. She's asking, what are the mechanisms to translate the tracking and indicators into political action?
Yeah, so the Landsat countdown has been very successful in engaging with policymakers. I believe the report was cited by the head of the UN as an example to be following. So it's very useful at sort of that high level decision making, because it provides very clear messages about the direction of the climate change impacts on health, monitoring across a range of different things. So we're monitoring impacts, adaptation, mitigation and co-benefits, economics and finance, and social and political engagement. So it has a very effective way of reaching those high-level decision makers. And when you're looking at perhaps more regional scales, that's why we have now our regional collaborations. We have the Europe project I'm leading, we have the Asia project, South America, and small island developing states. So that's an opportunity to really tailor those indicators with the key stakeholders. So we're particularly working with the European Environment Agency in our case, and making sure that we're producing indicators that can help inform their decisions. And the finer you go, of course, there's more different things that you need to take into consideration. And we've been looking to incorporate sort of downscaled indicators into decision support systems and early warning systems to be able to, for example, inform decisions at the city level.
Thank you. There is another question asking, what measures for controlling mosquito populations are being more effective?
Well, that's a little bit out of my area of expertise, but yeah, I mean, there's been lots of innovations lately. We know that the Wolbachia campaigns have been pretty effective at controlling mosquito populations, and there's been, yeah, different innovations in terms of insecticides and different traps, smart traps and things like that.
Mm-hmm. Okay. Another question, well, we have at least two more, but I think we need to finish with these two questions. The first one, what alliances are needed in the field to fight that threat?
What answers are needed in the field?
Alliances, yeah, alliances.
Oh, alliances, yeah.
Alliances.
Yeah, I think partnerships are incredibly important. It's the most important thing really to get this collaboration, these solutions off the ground. So it's really important to be linking, well, local decision makers, so particularly in the case of mosquito-borne disease, vector control teams with local town halls and decision makers, interacting a lot with the community, understanding, if they understand the risks of mosquito-borne diseases, to be able to minimize the risk of additional breeding sites, and to be interacting with scientists, modelers, climate change specialists, to bring it all together to have an integrated approach to building climate resilience to health.
Well, we have a lot of questions. I will go for the last one from César Muñoz. He asked, is there also a predicted risk of expansion of yellow fever based on your data? Excellent talk, thank you.
Yeah, well, some of the predictions are based on the suitability for Aedes aegypti, which can transmit yellow fever. Yellow fever, of course, is also confounded by vaccination coverage. So in terms of climate suitability, yes, there is an increased risk, but then it very much depends on the vaccination coverage in the place of question, because the vaccine for yellow fever, one dose is lifelong, and so it can be very effective if it's distributed equitably. Okay.
Thank you very much, Dr. Lowe. It's a pleasure to hear you. So now we need to move to the second talk. Welcome, Professor Kock, Richard Kock. Just a brief introduction about him. Richard Kock is a wildlife veterinarian, researcher, and conservationist. In 2011, he took up a chair in wildlife health and emerging disease at the Pathology and Population Science Department in the Royal Veterinary College in London, the UK. He has been engaged closely with the Evolving One Health initiative and in promoting wildlife and environment in the health agenda. He was awarded the FAO Bronze Medal for his contribution to global rinderpest eradication, but he also worked on a range of other infectious diseases, including pestes des petits rumiants, Rift Valley fever, anthrax, and other wildlife disease. So, Richard, Professor Kock, the floor is yours.
Okay, can that be seen okay?
Yes.
Yeah.
We can see the.
Jolly good. Okay, so thank you very much indeed for the opportunity to talk to you. So yeah, we're talking about pandemics. So I'll stick to that. Very interesting talk on dengue, and of course these are now very much endemic diseases, but pandemics really are something slightly different. So I'll just try and explain what it is first, and then talk a bit about what I call structural One Health. To frame this idea of prevention and what the future strategies are. I think people want to know, what can we do actually in this context? So generally accepted definitions of a pandemic are really an epidemic disease that spreads very widely in a population to some sort of ecological limits. In the case of the SARS coronaviruses, they were really global, so a global pandemic. Now, what are the principles of epidemic prevention? So we want solutions. How do we do this? Well, we have a number of options. We can prevent emergence of the pathogen that causes the disease, maybe. We can immunize, and obviously that's something now which is very much happening throughout the world for COVID. We can prevent contact. We can, let me just, sorry, go back. We can control or eliminate the source of the pathogen. But actually, to prevent a pandemic, that won't work, and that won't work. We know that because the pandemic that we've experienced, it took a while to get the vaccine. It was rapid, but it still took a long while, and we still had a lot of deaths. So the only thing really that we can do to prevent a pandemic is to prevent the emergence. So that's really where I'm going to focus. Just so you understand some of the epidemiology of some of these directly transmitted infections like the coronaviruses. We have this thing called R. You've heard it a lot now, I think, about this reproduction rate. And really it needs contact. So you have to, so it goes to another individual. It may or may not cause an infection, but ultimately with these highly transmissible infections, it rapidly moves through the population and you have a sort of a chain of transmission. But with all these things, they are influenced by a lot of intrinsic and extrinsic factors. So, it's a very dynamic thing. It can change. Even within one epidemic, you can get significant changes, both in the host population, but also the pathogen itself. And if we look at coronaviruses, we've had 3 coronaviruses in the last 20 years of interest and concern. And one of those is the Sarsko V1, which we had at the beginning of the century. And this didn't create a pandemic, just luckily, and partly because of some of the intrinsic factors about this particular virus. It wasn't quite as transmissible, but it was a close thing. And we had associations with animals. But actually, as to where the source was, we are still not 100% sure. We believe that the bat populations hold some of the ancestral viruses, but this is true for many diseases. But actually, we don't really know what the proximate sources have been. And we know that the virus can go to animals. And it's, it was a regional thing. The MERS coronavirus, Middle East respiratory syndrome coronavirus, we've actually worked out a lot more of its history and epidemiology. We know the camel is particularly important as a sort of reservoir of the virus. And it seems under industrial conditions, when we intensify, this leads now to a spillover. And this virus could also be a pandemic virus. It already has spread partially, but let's hope it doesn't, because we don't have a vaccine for it. And then we have the coronavirus, and I think you all know that. But again, we still don't know the source. Now, the thing about extrinsic factors in relation to pandemics, we just need to remember that it's about resilience. In other words, will your system resist the epidemic? Will it actually stop the epidemic growing? Or are the people resistant to the infection? Because that's also important if we want to prevent pandemic disease. Because infection is one thing, but disease is another thing. What is interesting about COVID, there's a paradox actually, that it's the most advanced countries in terms of health and medical investment that have been the most adversely affected. So there's a real paradox. It's a lesson for us. And these graphs, these are the Global Health Security Index and the JE, which is an evaluation of preparedness of countries and so on for disease. And it's all the developed countries. It's us in Europe, United States, and so on who've been adversely affected. So this paradox is really important for us to understand. So key parameters. Now, for a pandemic emergence, is they really need to be novel because, in other words, there's no immunity in the population. They can be zoonotic, they can be non-zoonotic, but they need to be that. They need to be highly transmissible. We need a population that's connected. Often plasticity helps because it provides secondary reservoirs, hosts, and so on. So the virus can sort of find a way when we're controlling it to keep going. Within a population, and this is really important, and I think particularly pertinent to this paradox with COVID, is issues like super spreaders and vulnerabilities, unhealthiness, if you like, and the close links with obesity and age shows why societies which are medicalized, wealthy, have been adversely affected. But other issues like poor collaboration, weak powers of intervention, humans are difficult to work with. And then we have structural drivers, pandemic drivers, like transportation, agriculture, political ecologies and economies. So it's a very complex story. So to prevent emergence, we've got to work across these sectors. Just a quick point on zoonosis. This is a very confused term. People use it and abuse it. But as a wildlife person, I just want to get across the message that if we look at, and on the left here, we have a big circle. The majority of zoonosis comes from animals close to us, and that is domestic animals. And of the wild animals that transmit infection to us, it's usually the ones that, again, we're close to. So peridomestic wildlife, like a rodent that shares our our environment, our housing, or a captive animal in a farm, perhaps a wild animal in a farm. Free-ranging wildlife is a very, very low risk. The thing about SARS coronavirus is that it's a zooanthroponosis, actually. We know that humans infect animals. What we don't know really is, do animals significantly infect humans? I think it's possible, and it's probable that we got it from an animal at some point, but actually it's more of a zooanthroponosis than a zoonosis, and that's something that people don't really understand. So what are the strategies to prevent pandemic disease at global and national scales? Now, I want to just, this is far too much text, I'm not asking you to read it, but just the principle of this pyramid. If we look at health systems, and you can look at the reference if you want, most of the investment in health is in emergency medicine, in medicalized society, taking pills, taking drugs, going to hospital, having surgery, having diagnostics. This is where 95% of all medical investment goes, and that's our health system. Our response, in other words, if you respond to these things, we don't really look that much at prevention. Vaccination is an exception. Since Jenner and smallpox and so on, it's been a very useful tool to prevent infectious disease events, because we create a natural immunity in the population. So that prevents emergence, prevents a pandemic. So vaccination is a really important part of, of our medical armory. One Health has tried to sort of expand this across environment, animals, people, to understand better the epidemiologies and to try and find ways to reduce risk, which is fine, but it doesn't go far enough. We need structural One Health, which is the lowest part of this, which is incorporating broader inputs, including deep time historical and cultural infrastructure and circuits of capital. It was interesting hearing the dengue talk, but for me, the real problem with dengue is obviously development related. And it's actually the investment by capital, by large wealthy countries in things like plastic and plastic materials, which then go into these slum environments that create opportunities for water to aggregate and gather. And it's that really that's a primary problem. If you could stop that investment, if you could stop that, if you like, attractant to mosquitoes, you will reduce these problems substantially. The idea that an urban environment is a safer environment than a deep forest, for example, when it comes to things like mosquitoes, is naive, because in a sense we generate that ecosystem. We fail to address things because the dominant political economy drives pandemic risk. It goes with the economic growth. So we get these paradoxes in vulnerability. So we need to rethink our political economy. Also, human-centric concepts of health and policies ignore externalities and drivers of pandemic risk. So we're very, very human-centric in the way we think about this. That's why One Health is a struggle, actually, to get people to think about the environment. WHO thinks about humans. OIE, which is the Animal World Health Organization, just thinks about domestic animals and economy of animal-based food systems. We don't really have a global wildlife health system or agency. So we never think about it. We never know how to deal with it. And we tend to speculate and pontificate about it, but we don't really know. So we don't have that investment. We're not operationalizing the One Health approach. And of course, in the context for pandemics, rapidly expanding human populations, but more important, massive domestic animal populations. 96% of mammals on Earth now are domestic. And so we create these conditions for pandemics and all the instabilities associated with the ecosystemic disruption. So world is one. Travel, massive, attractant to pandemics. Domestication, I've mentioned. Many human diseases are directly linked to domestication of animals. Modern food systems, massively increasing risk in relation to pandemic disease. We may find out eventually exactly how the SARS COVID-19 emerged, but it may be related also to farming of animals. So I mentioned about how domestic animals have really crushed nature and caused real problems. This biomass issue, even domestic pets, massive numbers, which also create problems. And if you look at pandemics, recent pandemics like H1N1, That did not come from nature. It came from a highly agriculturalized environment in Mexico. It was a pig farm, had four continents involved in the origin of the virus, from animal-based food systems and mixing with human viruses and ending up with a pandemic virus. So, some of these concepts around, like, Pandora's box and it comes from nature. No, it's a human-driven issue. It's and we need to recognize that and stop blaming things which really we don't understand. Similarly with H5N1 bird flu, related to the investment in poultry in Southeast Asia, massive growth in that industry, and then spillover of these viruses back into nature and into human populations. So we've got various opportunities. We need to shift from this focus, stamp collecting of viruses, etc. We need to move on to really important, I think, social and economic changes, reduce air travel risk. We need to refine the Global Health Security Index. We need a healthiness index, actually, in our societies, so we know what our resilience will be to pandemics. We need to shift public health measures towards increasing this resilience, and we need to modify a lot of our systems. So big changes are needed in this context. And I can go on. I mean, we really need to change, and I think young people recognize this. Unfortunately, many old people just continue to, as some people say, blah, blah, blah. But thank you very much indeed. Enjoy the opportunity to talk to you.
Thank you very much, Professor Kock. We can start with the questions in the chat. So I'm going to start with the first one. It says, is the higher risk of elderly behind the paradox of higher death rates in rich countries? Could you comment on other potential scenarios in other potential pandemics?
Yes, yes, of course, age has been, an important factor in this, and in the mortalities, and it's biological. I think we just must understand. I mean, it's great that we can live longer. I'm very grateful for be able to be alive at my age. So I don't think the issue is age particularly, other than we have to protect that age group. They're very vulnerable. In this modern world now, where we are such an attractant to pandemics. There are going to be more. I mean, if people think this is a one-off, sorry. I mean, we've had 3 potential pandemics from this family of viruses alone in 20 years. So I think it's very likely we'll get more. The question now is, how can we rapidly, increase our resilience to this? So the first thing is to recognize our vulnerabilities. We have this tendency, particularly Eurocentric communities and cultures, to think we're right all the time and to impose our ideas on other people. And many of them medicalized and based on profit from pharmaceutical companies. Sorry to say that, but that is true. That is part of the economic model, and it's what makes countries rich. But we need to reevaluate that and say, well, actually, we need to look at the grassroots of these things. And that's why, for example, obesity is directly linked to actually social determinants, discrimination in society, but also poor availability of food of good quality. So, and that is a profit issue. We make food, which then causes obesity in people. Yes, people have some responsibility, but actually they don't have the choice. And so they have, highly calorific foods and so on. So I think there's a lot at the social and economic level that will prevent pandemics. That's where we need to work, rather than getting wrapped up in what comes out of bats. Thank you.
Okay, thank you. Another one. In your opinion, is it better to reduce overpopulation both in humans and animals, or to have a better control of their health?
Well, of course, population is a key factor in pandemics. So, if we have a big population, we are going to have pandemics. So you just have to recognize, and this is just a fact of life now. I think population of humans will stabilize. It doesn't worry me so much, and I think it will probably decline over the century. I think people again are rationalizing in the context of resource limitations, etc., and family sizes are dropping quite rapidly. So I think we'll resolve that, but we are going to have a difficult century because we have so many people. What we could do, and I think it's a very easy thing to do, but sadly not discussed at high level, for example, at COP26, I believe the lobbying from the food industry kept the issue of animal-based food off the table, sadly. These are powerful lobbies, but it's really simple. We know we could reduce meat-based and dairy-based food by probably 75% without any big economic impact, and improve health and reduce risk from this large domestic animal population, which, as I said to you, is 99% of all zoonoses, all diseases humans acquire from animals directly, come from the domestic animals. So you could, in a stroke, really resolve that. But of course, there are many cultural issues. There are people who live in high mountains, in deserts. They can't survive off crops easily unless our distributional networks allow it. So some real challenges, but it's such low-hanging fruit, this, to change our diets, human diets, and get all the health benefits and the healthiness, which increases the resilience. So whatever, if you like, nature, throws at us in terms of microbial challenges, we'll have resilience and resistance. Look at the difference between the African continent, where we have a lot more resilience in the population there in terms of COVID. It's a complete contrast with Europe and North America. I know it doesn't cheer up our politicians, because now they've got to try and justify, exactly how we've developed, because we like to say that Africa is underdeveloped. But we need to rethink these things and rethink health in this context. Thank you.
Thank you. Well, we have many more. I need to choose only one, and I will go by the first one it appears. So, could you elaborate on the concept of plasticity or secondary reservoirs? Does this capacity depend on the type of pathogen or available high numbers of secondary reservoirs, pig, poultry, etc.?
Yeah, so plasticity, this is really a viral characteristic. So RNA viruses, for example, they are able to invade cells. There is a phylogenetic, that means a relatedness between animals as to whether the virus may infect one animal or another. But it's just a characteristic, and obviously it's valuable if you're a virus to do that, because you need a host. Unlike many bacteria or parasites and things that can live outside the host, viruses, they need a host. So that plasticity helps in a sense. I'm using terms which are a bit anthropomorphic, but in other words, it's better for the viral population if it can do that. So yes, in a sense, if you have a big domestic animal population, which you're dependent on, and you have a big human population, the two will act together. So these viruses will be able to move between them. So the evolution, look at measles. Measles came originally from rinderpest. You mentioned that in your introduction. And so it came from cattle. When we domesticated cattle, measles then emerged. From there, canine distemper came, which then has caused many problems around the world in carnivores. And then from there, we got phocine distemper, and we got cetacean morbillivirus. So, it all starts 10,000 years ago. We started this chain of events through our development, and we need to recognize this. And, it can take 10,000 years to get to that point. And it's an ongoing process, but it's us, it's humans who generate the conditions for this. Thanks.
Okay, thank you very much. I think now we need to move to the third talk. Thank you very much, because it was really interesting, the discussion and the presentation. So we now move to Professor César Muñoz-Fontela. He's the head of the Virus Immunology Working Group at the Bernhard Nocht Institute for Tropical Medicine in Hamburg, Germany. His career is mainly focused on immunology of hemorrhagic fevers, mainly filoviruses. And since February 2020, he's seconded to the WHO blueprint. He's the chair of the WHO Working Group on COVID-19 animal models. And he's gonna talk about the epidemiology and ecology of Ebola virus. So Cesar, the floor is yours.
Thank you very much, Julia, and thank to the speakers. I mean, to the presenters, and for the invitation to this very timely meeting. So I'm going to discuss with you today some concepts on Ebola virus epidemiology and ecology. The first thing I would like to say is that when we talk about filoviruses, most people focus on Zaire Ebola virus, which is the cause of most of the outbreaks. In particular, the most famous ones was the West African outbreak, but there's other Ebola viruses, and of course Marburg virus as well. And some of them are also highly pathogenic for humans. We're talking about, for example, Sudan Ebola virus, Bundibugyo, and probably Typhores, and some others are very poorly characterized. So the first known Ebola virus outbreak was caused in Jambuku, what was at the time Zaire, and is now the Democratic Republic of Congo in 1976. Since then, we've had also some other outbreaks, smaller, and I'm talking about 300 people with very high case fatality rates, about 90% at the time. And then we had also smaller outbreaks in the western part of Central Africa, in Gabon, in the Republic of Congo. And as you probably know, there was this big epidemic in West Africa between 2013 and 2016, which caused to more than 20,000 cases and more than 11,000 deaths. And actually, since then, we have had Ebola outbreaks essentially every year. And we still have an unresolved small outbreak in DRC, which means that probably because of the increased surveillance that came after West Africa, we are finding more things, because obviously if we are looking for things, we will find them. So Ebola virus is supposed to circulate in bats. We don't know which bat species. Nobody has ever found infectious Ebola virus yet in a bat, but people have found antibodies in different bats, and some bats were also PCR positive. So people have found genomes, but we don't know which kind of bat. But what we know, there's other species also that are very highly sensitive to Ebola virus infection, like forest antelopes and apes. One thing that is important about the Ebola virus is that epidemics start with a single spillover. Can be, for example, a hunter or somebody that is in contact with one of these intermediate reservoirs. And then all epidemics are fueled by human-to-human transmission. And this is very important, and this is very different to any other viral hemorrhagic fever, which usually depends on the presence of vectors, or they are, for example, multi-spillover events. But the Ebola virus is not a highly transmissible virus, but it's highly infectious. And then there's three situations in Africa where typically Ebola spreads. One is in the household, and this is one of the reasons why women are usually more affected than men. There are more cases of women because they are primary caretakers in the household. In the nosocomial settings, so in the health settings, and this was very typical of the first outbreaks through needle stick injuries and traditional funeral practices. And one thing that we know now, and I can promise you that has changed completely, everything that we thought about the Ebola virus, is that the virus can persist for very long periods of time, and we're talking about years, in survivors, in immune privileged sites in survivors like semen, for example, or the central nervous system. So we have had recently outbreaks that had started from a survivor via sexual transmission, for example. So in some settings, in a way, we don't need a zoonotic reservoir anymore. The risk factors that are responsible for spillover of filoviruses are not so well known, but for sure frequency of human-animal contacts and the presence of reservoirs and intermediate hosts as well. The pre-existing immunity in human populations is not very clear. Probably most people are immunologically naive, but there's situations that in people that live in rainforest areas that have antibodies against Ebola or maybe some other unknown filovirus. global climate change, as the first speaker was mentioning, is very important because it's changing the bat migratory patterns, for example. So now we find bats that usually used to migrate to DRC in higher latitudes. And as I mentioned, human reservoirs. And sometimes in the case of Marburg, I can tell you that human stupidity is also a very important factor, as you can see here in this picture. So regarding the filovirus host range, is not really well known. There's, for example, some hints that that pigs can act as an intermediate reservoir. But the bottom line of this slide is that after each one of the outbreaks, people have formed, people like expeditions were mounted to try to find reservoirs, to try to find the virus in nature, and nobody has ever found anything. So right after West Africa, my lab, like a lot of other people, did some bat trapping in the areas where the epidemic started. This is in Gekedu, this is at the border between Guinea and Sierra Leone. And I can promise you that we also didn't find anything, but one thing that we found is a pseudovirus. This is a remnant or a paleoviral sequence, if you want. It's a virus, it's a filovirus derived pseudogene that is inserted in the genome of this bat, Mops condylurus, which is also it was related with the presence of another Ebola virus called Bombali Ebola virus. So this is not new. It is known. It's been known for some time that filoviruses can integrate in the genome of the host through non-homologous recombination with mobile elements in the cells, like transposons, for example. Sorry, apologies. And we know that rodents, bats, and marsupials can have some of these filovirus pseudogenes. But what is interesting is, for example, the one that we found, which is here called NIR15, has the closest identity, the closest resemblance to the actual Ebola virus nucleoprotein. It's inserted in the genome, and with this sequence, together with other sequences that other people have found, indicate that filoviruses are ancient, are really, really old. So the most common recent ancestor between true filoviruses that are here, as opposed to these pseudogenes, is more than half a million years ago. So just a short take-home message from this part. Basically, all these all these filovirus pseudogenes found so far are derived from bornaviruses and filoviruses. So they belong to the order Mononegavirales. So there's something about these viruses that allow them to integrate at some point parts of the genome in the genome of the host. These integrations are generally limited to one copy, which suggests that additional integrations confer no advantage to the host. In the case of filoviruses, all these NIRs are derived from the nucleoprotein or BP35. This is another viral protein. So essentially, from the whole viral genome, which is 18.5 kilobases more or less, only pseudoviruses are formed from the nucleoprotein or BP35. The conservation of relatively long open reading frames and a potential correlation between NIRs and resistance, because we're talking about bats, rodents, and we don't know about marsupials, but for sure bats and rodents are resistant to filovirus infection. So this is consistent with some biological advantage. And finally, a message for you today is that filoviruses are really, really old. Changing gears a little bit and talking about the epidemiology, one of the things that we are still trying to understand is why, in the case of West Africa, we went from outbreak to epidemic. We didn't reach the pandemic stage, but we went to epidemic. And I think molecular epidemiology has really helped to understand that, because now we are able to sequence viral genomes from patients more or less in real time. And that, looking at the little variations, little mutations that appear in these genomes, we can track how the virus spreads in the community. So by doing that, I'm going to show you how the virus spread from the epicenter of the epidemic in Gekedu. So this is Guinea, and this is Liberia, and this is Sierra Leone. And I'm going to show you how the virus spread during this epidemic. So you can see this is going to be important. This is Kenema in Sierra Leone. And this was the source, for example, of some traditional funerals. And you see how the virus really spread across the borders of the three countries. Now it's touching the capital. This is Conakry, this is Freetown, and this is Monrovia. So you can see that movement of people was one of the main drivers of the epidemic. So multiple cross-border movements of virus. That was one of the main drivers, as I said. Doing molecular epidemiology, we also can infer that the origin of the epidemic was probably a 2-year-old child that died in December 2013 in Meliandou. This is a village of Gekedu in Guinea. And this is just to show you that when we were doing sequence in the field, this is a graph that shows the time, the date the sample was collected in red by the European Mobile Lab in this case, and the time the sample was sequenced. So you can see that in most of the cases, the samples were sequenced at the same day that the samples were actually collected. So I think this was done, and by the way, using nanopore MinION technology, and I think this was probably important to follow and to control the epidemic, at least to some extent. I'm going to give you two examples of how molecular epidemiology helped to control the epidemic. In 2016, we didn't have transmission anymore in Guinea, but all of a sudden we have two new cases, or actually spots of transmission. One was in Boké and the other was in Siguri. So by doing molecular epidemiology, what we actually found out is that the case in Boké was from Koya, so somebody who traveled from Koya into Boké, and the new cases in Siguri was actually a reintroduction from Sierra Leone. The other thing that was important, and this is another example, this was a case in 2016 also when there was no transmission of a person, a female that died from infection, and she actually before dying, actually unfortunately she passed the virus to her husband and then some of her children. And then what we found out is that her virus was essentially similar to the virus of a survivor. So this was a case of sexual transmission, and the survivor was her neighbor. And I would like to finish the talk by, we discussed a little bit the One Health concept. This is one of the examples why I think social science should be also included or taken into consideration in the One Health concept, because there's an important stigma now of the survivors. I think in some ways it's comparable to the early days of HIV, where they are expelled from their communities and they are considered as a source of the virus. And there's a lot of important work that we need to do in this regard. So just as a take-home message, the Ebola virus outbreaks are caused by single spillovers from a zoonotic source or via sexual transmission. Molecular epidemiology substituted classical epi and proved essential for epidemic control, and the population mobility is a main driver of Ebola transmission. And I would be happy to take questions, and thanks again for the invitation.
Thank you very much, Cesar. I'm gonna start with the questions in the chat. So the first question from Nuria Izquierdo Useros is, what are the possible mechanisms allowing Ebola virus to persist for so many years in humans? Or how can these reservoirs persist for so long?
The short answer is we don't know. I think it's going to change. I mean, we don't even know if it's persistence, if the virus can produce some sort of intermediate DNA elements and persistence and episome. I mean, we are really considering, we are actually having a talk very soon, a meeting, and people are considering all kinds of situations, but the mechanism of persistence is not known. What is known from experiments in non-human primates is that resident macrophages at these immune privileged sites that are CD68 positive are the ones where the virus is found. So that's what I can tell you at the moment.
Okay, thank you very much. Next one is viral persistence in survivors related to integration of the virus or to stability of viral particle in immunological sanctuaries? More or less, yeah.
Yeah, again, it's really, I mean, I think somebody is going to find something very soon because there's a lot of people looking into it. We are partially looking into it as well, but the mechanisms are not known at the moment.
Okay, next question. Why are epidemics increasing their frequency and number of people infected in the recent decades?
So I think that there's two factors. In the case of West Africa, the communications, so the road, the infrastructure is much better, for example, in West Africa than in Central Africa. And this was the first epidemic where everyone had a cell phone. So now, you know, in the old days, if you want to invite, if a relative dies and you want to invite a person to come to the funeral, maybe that relative cannot come or doesn't know in time, you know. But now with the cell phones, everybody is informed, and the roads are much better, so people can travel. And the reason why we see more outbreaks is simply, I think, in my opinion, because there's a much better surveillance network. So now there's more diagnostics. People are looking into what is going on, and people are finding things, which probably means that in the old days, nobody really knew if the virus wipes out a small village in rural Africa. You know? It's difficult to know.
Okay, there is another one saying, if filoviruses are not transmitted by air, how is mobility having such a great effect on epidemics?
No, this is because, of course, the virus travel with people. So people, you know, the political barriers between Sierra Leone, Guinea, and Liberia are political, but there's different types of tribes, the Fulani, the Kisi, the Malinke, and they cross these borders because they are the same people. So they are used to this. So basically what we have is a lot of people infected going around and meeting other people, or going to a traditional healer and then going to a traditional funeral. And then, you know, there was a lot of super spreading events in traditional funerals, so that people go, and traditional funerals, the body is washed and there's a lot of contact with the body, and then they go back to their places of origin. So that's another way how the virus was spreading.
Mm-hmm. And in fact, I think it's very important. All of the speakers mentioned, no, this One Health concept, and sometimes we only think about the triangle, no, like the human health, animal health, and environmental. And here also come indirectly other things, such as communication. I think is very important, but also taking into account in which country, which traditions, which everything involved, just to either to prevent, to treat, or to do some actions to fight against this thing.
And the message from the leaders, the message from the authorities, which is something that we are also seeing in the COVID pandemic, is very, very important, I think.
Exactly. Okay, I will go for the very last one because we are out of time. Saying, how is the uptake of the novel Ebola vaccine in ring vaccinations strategies? Has it helped to control recent outbreaks?
Yes, it has helped a lot for two reasons. Because people really want the vaccine, and which is something that probably we should learn about. And then the other thing is that the people that are doing the vaccination on site are very experienced, and the contact tracing is working very well. So it has really helped to contain the latest outbreaks. Yes, absolutely.
Okay, thank you very much, Cesar. I think now it's time to close the session. I hope you all enjoyed this session as much as I did, because it was a very big pleasure to moderate this one with such amazing speakers. And in fact, I want to thank all of them for their nice talks, and also the organizer, as Cesar said. No, it's very important to set up this this type of sessions, talking about zoonosis, about One Health, and the importance, that is not only to talk about public health, but we need to combine it together with animal health, with environmental, and importantly with politics, with communications. So yeah, thank you very much to the speaker, organizer, and all of you to attend this session.